Okay, if you're doing a collection in a farmer's field, you want to discuss with the farmer what varieties are planted where, because there could be more than one variety in the field. So for instance here, I can tell I've got one variety. It looks very different than, say, the other variety next to it. And insofar as we're taking measurements of the field in terms of the space covered by variety, you need to make sure that you measure those different areas based on where the variety is. Now for corn, usually those will be planted separately, but for some other crops they might be intermingled, and that becomes difficult. You'll have to measure the whole area and then sort of look at what percentage might be one variety or another. But often growers, farmers will separate the different varieties, so it's really just measuring one globe or area and then the other. So you'll want to measure that if you're measuring it. Otherwise you might be using a farmer estimate or an indication of how much they planted in order to estimate area. Then the second thing is collecting a sample. So if you come in here and want to collect a sample, depending on your crop and the time when you actually get to that crop, there's different material, plant material, that you might want to sample in order to be able to get an understanding of the genetics. So here we have a crop that's already set an ear, and you can see all of the tissue here is pretty dry. Even the green tissue is really old tissue, and so there's not really good, it's more difficult to do DNA extraction on old desiccated plant material. And so your option really here is going to be to harvest kernels from the ear. And so the other thing to consider is when you're harvesting kernels from an ear, how many plants are you going to sample? And then to make sure as much as possible you're not sampling them all from just the same little area. The reason for that being is that even though it might all look like one variety, it is possible that there's other things going on there that are kind of spatially segregated. And so you'll want to sample throughout the area that is indicated as variety. Often you do that as a W shape, which is something that in ecological sampling they found works well. So you'd come in and you'd sample some grain off of one ear. You put that into your sample bag, and then you'd walk into the plot and you would sample grain off of another ear. And you can see here, you know, when you're sampling, there can be molds and all sorts of other things. Those can affect the ability to extract quality DNA, and so you'd, you know, look to try and sample kernels that aren't so damaged like that.